I want to marry for working. Yeah. You can like choose that. You can put it on YouTube for everyone or not. Yeah.
Yes. All right, well, we're going to start 1 minute early because we have a great turnout. I'm going to go ahead and do Thursday really fast. Very good. So this will be quick, but we have two pieces of good news from Dr. Vera Potter today. First off, a previous McNair research scholar, Eric Kasem, had a paper published called Ketogenesis Supports Hepatic PUFA Homeostasis via Fatty Acid Elongation, and published in Science Advances, so congratulations to your lecturer for that. And then on top of that, Dr. Vera Potter and Dr. Paley's RO1 got scored 34th percentile, so congratulations. So with that, I'll pass it over to Dr. Rector.
I didn't mean to overstep. Thanks for coming today. That is great. So I heard ketogenesis in there somewhere, right?
Appropriate.
Yeah, it's very appropriate. Great transition for the topic today. So I'm happy to introduce Grace Shryack. All of you know her. She has a slide or two about her history and how she ended up here. I'm not going to navigate that for her, but Grace has been with us since 2021, right? So we were right in the figuring out of the pandemic. She was very instrumental when she first got here based on her human exercise physiology background of helping Dr. Parks and I wrap up an RO1 where we actually took ellipticals and stationary bikes to people's homes because we couldn't exercise in facilities here technically at that time still. But Grace has been fantastic. This is not her dissertation topic, and she will present that in a few months, but this is other things that she's been working on in the lab. She's been very productive, has one co-first author publication, a second first author, another first author publication that is in revision right now. Hopefully we'll resubmit within the next week or two. Co-author on 4 other manuscripts in the lab, and has another one that's on my desk as another first author publication. So very excited with what she has been doing and the progress that she's made over the last 3.5 years. And so she's going to tell you a story about some of the work that we've been doing in the lab.
So I come from a small town in Illinois called McClax for you. After I graduated high school, I moved to Edwardsville, Illinois, just down there, where I attended my undergrad and my master's institution. I worked in St. Louis for a few years, and then I hopped on over to Columbia, Missouri. For those that were here for Brian's presentation, I know my map is much more zoomed in than his, clearly, but I will say I always knew the direction and the state that I was going to, so. So I attended my undergrad at Southern Illinois University, Edwardsville, as well as my master's. During my undergrad, this is when I first became exposed to research, specifically exercise science research. I met this man with the red arrow, Dr. Brian Smith, and he at the time was doing research related to smartwatches and prediction of VO2 maxes. So we're able to put individuals on VO2 maxes and compare it with smartwatch predictions as well too, along with some other fun projects that includes DEXAs, et cetera. After I graduated in 3 years. I was 21, and I was like, you know what? I don't want to make any big girl decisions yet. What should I do? Cool, they'll pay for me to continue doing what I like to do. So I'll get my master's, I'll become a GA, fantastic. I was the only female GA in the department at the time, but I had a lot of fun. You know, I was able to really learn a lot about myself teaching, still participate in research, and I was able to complete this in about a year and a half. At the time, I thought, you know, maybe I'll go to PT school, maybe I, you know, would go down a different path. I was ultimately undecided, so I decided that I would work in St. Louis for a couple of years and, you know, try to figure out what really my true interests were for a career. So I was a personal trainer and a gym manager at Catalyst Strength and Conditioning in the University City, Missouri, which is in St. Louis. It was a small local gym, but we had a wide variety of clients. You know, we had blues players, we had young children with intellectual disabilities, we had just our local community guy, 75-year-old guy who's executing a perfect Bulgarian split squat, I must say. I trained him myself, thank you. So I was having a lot of fun in this role and I was excelling at it at the same time, but there was aspects in my career that I knew I was missing. I really missed teaching and I missed research and I really missed that education aspect and, you know, being inquisitive and asking myself questions. So I contacted this guy again and I said, "Hey, what should I do with my life? Do you have an idea? You know me." And he said, "I think you should get a PhD." And I was like, "Oh, okay, you think I'm smart enough to do that? Okay, cool. Let's try it." So I actually applied to PT school and PhD school at the University of Missouri because I thought, you know what, I'm good at the personal training thing. Maybe if I'm on the front end, I'll enjoy it a little bit more. But ultimately, I ended up deciding to do a PhD for a number of different reasons. I won't dive too much into that because we'll be up here all day. But now my camera roll looks like this. So it's buffers and primers that need to be ordered, Western blots that you're like, yes, this finally works. So I must commemorate this moment. Old lectures that I found incredibly interesting, but will probably sit in my camera roll for the rest of my life. And some of my really good friends that I've been able to attend conferences with. So here I am almost 4 years later. And as much as I love to hear my own self talk about myself, let's go ahead and dive into the research a little bit. So a quick outline, we will be discussing ketones, mazold, how these two things relate, ketones in the brain, and then ketones in exercise. A quick little disclaimer, NAFLD and mazold are the same thing, so non-alcoholic fatty liver disease and metabolic dysfunction associated cirrhotic liver disease, mouthful. It's the same disease, it just recently has been updated with a new name, but I will use them interchangeably throughout this presentation. So first, what exactly are ketones? They're a molecule in our system made up of hydrogen, carbon, and oxygen. They're typically derived from fatty acids from our adipose tissue. They're considered an alternative, I put these for a reason, alternative fuel source, because they're increased utilization during times of fasting, starvation, pregnancy, and exercise. They're produced by hepatocytes predominantly, which is a cell type in the liver, but there are other tissues that can produce small amounts of ketones, such as the kidney or your intestine. They're utilized by a number of tissues, including the nervous system, heart, skeletal muscle, brain, kidneys, and this list is slowly increasing as we start to learn more and more about ketones and their function. The human liver produces roughly 300 grams of ketones a day. This accounts for 5% of our total energy expenditure when we're in a fed state. So when we're eating, we're using glucose, carbs, we're using fat. We don't need a lot of ketones to be produced, so it's a smaller percentage. Then when we stop eating around, 8 p.m., we go to sleep, we wake back up. We have not consumed any food, so ketones are naturally going to be increased. It's sort of the circadian rhythm aspect of ketones. On the right-hand side, those are our three main ketones in circulation. Beta hydroxybutyrate, which is the most prevalent one, exists in 70 to 80% in circulation of our blood concentration for ketones. Acetoacetate is our second most prevalent, which is closer to 20%. And then acetone, which typically gets excreted off pretty quickly, is in 1 to 2%. There is exogenous and endogenous ketones. So what I mean when I say this is ketones that come outside of the body that you can ingest, such as ketone esters, ketone salts, and endogenous. Most of my work, well, all my work so far, is surrounding endogenous ketones that are produced by the liver. So there's a long list of potential ketone benefits. Ketones are sort of a hot topic in science, and more literature is coming about coming out about the potential benefits that ketones can have on many different tissues, some of which include heart failure, gut health, feeding behavior, energy expenditure, Alzheimer's disease potentially. I just found an article the other day that they found that ketones could be beneficial post-concussion for TBIs, so very interesting. And the topic of today, MASLD. Ketones are commonly thought of as a metabolic substrate, and that's, they are, but they can also be utilized for other things like cellularly, including modulators of inflammation or oxidative stress, drivers of protein modification, and signaling mediators. On the right-hand side, I provided a quick table about what is the blood ketone concentration during different states. In our fed state, it's typically less than 0.5. And as we start to increase that fast, so we're not intaking any food, we see an increase. During prolonged exercise, it's up to 1 millimolar, and this can be dependent on when the last time somebody ate, the intensity of the exercise, the type of exercise, male versus female. So there's a lot of different factors that can change your blood concentration of ketones. Diabetic ketoacidosis can go up to 20. And then I also wanted to highlight pregnancy. During different stages of pregnancy can be slightly elevated from baseline. So ketogenesis, or the production of ketones, occurs in the mitochondria of the hepatocyte, which is in the liver. What happens is we get these fatty acids taken in by CPT1, gets broken down by beta oxidation or fatty acid oxidation. From there, we get ACAT1, which then breaks it down further, and we get to our rate-limiting enzyme of this pathway, which is HMGCS2. Following HMGCL then yields us ACAC, which is our second most abundant ketone, or acetoacetate. Acetoacetate then can be converted into beta-hydroxybutyrate by BDH, or acetone can be given off slightly. MCT1 is the transporter that will export and import our ketones, whether it's beta-hydroxybutyrate or acetoacetate. Once it's taken into another tissue, we get ketolysis, or the breakdown of ketones. So we take in our BHB, it gets broken down by BDH, we get ACAC, and then SCOT, or OXCT1, further breaks it down, and then ACAT1. If you notice, some of the enzymes are similar between our ketogenesis and our ketolysis pathway. So these enzymes can work in either direction, such as ACAT1 or BDH, and our transporters is the same for MCT1 as well too. So at this point, I hope you're thinking, okay, you're gonna talk about the liver. Can we get to it, maybe? Well, what does it have to do with MASLD? Let's back up for a second and let me tell you a little bit about the disease. So in a beautiful, healthy liver, we get these really nice looking hepatocytes. They're nice and organized, the nucleus is in the middle, and we have small droplets of fat. NAFLD, or MASLD, occurs when we start getting greater than 5% of lipid that is present. So we get these large droplets of fat, our hepatocytes become a little disorganized, we get some bloating, and our nucleus becomes displaced. We then get to the progression of this disease, or NASH or MASH, and what happens is we start getting increases in inflammation, we get dying hepatocytes, we start seeing increases in oxidative stress, and we start getting collagen fiber formation. Cirrhosis, or what's considered sort of the end stage of this disease for the most part, we have a very sad looking liver. We have lots of scarring or fibrosis. We have lots of remnants of cell death. We still have a lot of lipid, and we have a lot of inflammation and oxidative stress. We have a very poor functioning liver. There's not one main reason as to why this disease occurs. It's considered a multi-hit hypothesis. So a lot of things can affect the liver. We can have insulin resistance. We can have dysfunctional adipose tissue. We can have an increase in adiposity. We can have poor physical fitness. We can have intake of certain nutritional factors. I mean, sleep apnea even affects the liver. So there's a lot of things going on that can affect the liver. But what we do know, and a lot of experts agree on, is that the mitochondria is the cornerstone of this disease. So our mitochondria is responsible for a number of different processes, metabolic processes specifically. So we got our fatty acid, it comes in by CPT1, like I mentioned before, gets broken down by beta oxidation, or fatty acid oxidation. It leads to the TCA cycle, and then eventually into our oxidative phosphorylation, our complexes 1 through 5, to give us ATP. So some major pathways going on here. At the same time, the mitochondria is also going through elements of turnover. And what I mean when I say this, if you think back to like when you were in 5th grade and you had to make like the mitochondrial cake and the different stages with fission and fusion, biogenesis, autophagy, mitophagy, it has this natural self-check system that is, for a broad term, turnover. What also occurs in the mitochondria, if you remember what I just talked about, is ketogenesis. So when we get to this masoled state, what happens is we get a decrease in beta oxidation, a decrease in our TCA cycle, our decrease in our oxidative phosphorylation through those complexes, and a decrease in our turnover. Mary Moore, who was a previous, Dr. Mary Moore, was a previous PhD student in Dr. Scott Rector's lab, actually looked at a few different elements of this in humans. A lot of the work previously was done in rodents. She was one of the first to really show a lot of this information. I won't dive too heavily into it, but I really just wanted to highlight, as our disease progresses from healthy to more end-stage NASH, when we're looking at fatty acid oxidation or beta oxidation, I think you guys can appreciate this decrease in all of these. And beta activity, beta hat activity is an enzyme that's a part of fatty acid oxidation, is decreased as well too. Similar with our mitochondrial biogenesis markers, we get decrease in these. Our mitochondrial dynamics markers, again, relating to turnover, decrease. Autophagy markers, decrease as well too. So at this point, I hope you're thinking, well, you didn't talk about ketogenesis yet. I haven't. So all of these things are decreasing in masolds, right? We get a decrease in fatty acid oxidation and these other metabolic pathways in turnover. In healthy individuals, we know that fatty acid oxidation occurs in rates proportional to ketogenesis. So essentially that means that fatty acid oxidation should essentially equal how many ketones, for the most part, we have in circulation, right? As well as some of the enzymes. In masolds, up until recently, nobody's really evaluated whether this decrease in fatty acid oxidation occurs proportionally to the ketogenesis. Not only this, but there is some literature where individuals use ketones as a surrogate marker for fatty acid oxidation. So there's a true need to know, is there any sort of relationship between these? Some other work in our lab that includes a ketogenic diet and ketone ester. Just once again, I wanted to highlight that there is some sort of relationship going on between ketones and whether it's hepatic metabolism or histological outcomes on NAFLD. The ketogenic diet and a dietary ketone ester were beneficial on the liver. Which then leads me to one of the first pieces of work that I've been able to be a part of here at Mizzou. So we looked at the relationship between serum beta hydroxybutyrate, our most prevalent ketone, hepatic fatty acid oxidation as well too, and a few other mitochondrial markers in individuals with obesity and NAFLD. So we were able to take liver biopsies from individuals undergoing bariatric surgery just over here in the School of Medicine in the hospital. And then we were able to do H&E staining, which makes up a NAFLD activity score, which I will discuss here more in a second. And then we're able to look at fatty acid oxidation, beta hat activity, circulating ketones, and then we did some protein and gene expression with some mitochondrial markers and our ketogenesis markers. So when we get our liver, we typically do H&E staining and trichrome staining. H&E staining allows us to look at how much fat is in the liver. So these white droplets are fat in the liver. Trichrome staining allows us to look at fibrosis, and these purple lines tell us how much fibrosis is forming. So when we evaluate NAFLD's activity score, or NAS, there's 3 main components that we are looking at. We're looking at steatosis, or how much lipid is in the liver, inflammation, and then ballooning. So that bloated hepatocyte that we looked for before, that's what ballooning is. So we have a grading score of our NAFLD activity score, with 0 being absolutely no disease. We can look up here. It's a nice, beautiful liver with just a few droplets of fat. 1 to 2 is mild disease. We can hear we're starting to get some sort of fat formation. 3 to 4 is moderate disease. We can start to see a lot more fat. We're starting to get a little bit of inflammation as well too. And then a NAS of 5 and greater, I think you can appreciate how much more lipid is in that liver, as well as this purple lining, which is fibrosis as well too. Fibrosis does not make up a part of our NAFLD activity score, but it is something that we do pay attention to. So we were able to evaluate HMGCS2, which is that rate-limiting enzyme of ketogenesis, in our mRNA expression, our protein expression, and we evaluated it by our NAFLD activity score. And I think you can appreciate that as our disease increased, this expression in mRNA and protein significantly decreased. HMGCL mRNA, which is that enzyme just below HMGCS2 in the pathway, insignificantly decreased, but it was trending. Beta hydroxybutyrate, our most abundant ketone, significantly decreased as that disease increased. So ketogenesis markers decrease as our disease increases. We then evaluated by fibrosis, with fibrosis 0 to 1 and 2 to 4, 2 to 4 being a higher amount of fibrosis. And we evaluated the same markers, so HMGCS2 mRNA, protein expression, HMGCL mRNA, and beta hydroxybutyrate. HMGCS2 mRNA was the only one that was significantly altered. For all our other markers, there was no alteration. So overall, ketogenesis is not impacted by fibrosis formation. We then developed this heat map to look at the correlation between our ketogenesis markers, so HMGCS2 mRNA protein expression, HMGCL mRNA, and beta hydroxybutyrate. And we looked at that compared to our 3 elements of our NAFLD activity score, and fibrosis. As we start to get closer to a -1, we know that this means that there's an inverse relationship. So as steatosis or those lipid droplets increase, BHB decreases. So if we look just on our BHB serum, we can see that BHB has correlations with steatosis, inflammation, ballooning, NAFLD activity score, and fibrosis, similar with our HMGCS2 protein content. So again, suggesting that these 2 ketone markers decrease as our NAFLD activity score increases. We then evaluated 1 element of fatty acid oxidation, and we looked at that again with HMGCS2 and serum beta hydroxybutyrate. And we saw this positive relationship that as fatty acid oxidation increases, so do our markers of ketogenesis. So complete fatty acid oxidation correlates with ketogenesis markers. Next, we were able to look at mitochondrial beta-had, which if you remember from before, this is an enzyme that's a part of fatty acid oxidation, and CPT1A, which is that shuttle that brings in our fat into the mitochondria. And we looked at that in relationship to HMGCS2 and serum beta hydroxybutyrate, and we saw that these have a positive relationship between the 2. So elements of fatty acid oxidation correlate positively with our ketogenesis markers. Following, we evaluated different markers of that mitochondrial turnover that I discussed previously. So biogenesis, autophagy, mitophagy, and again, we correlated that with HMGCS2 and HMGCL mRNA. And as we get closer to 1, that indicates a stronger correlation. And I think you can appreciate in humans too, that's a strong correlation. For anybody that is human research, that we know that's a pretty strong correlation. So again, just once again showing that ketogenesis markers, they correlate with mitochondrial turnover, so the health of the mitochondria. And finally, we evaluated beta hydroxybutyrate lysine. So like I discussed previously, ketones can have other responsibilities besides metabolism. So this includes beta hydroxybutyrate lysine. So what happens is beta hydroxybutyrate attaches onto the lysine of a histone, which is part of the DNA structure that can be formed to make a chromosome. Hope I haven't lost you guys yet. And then what happens is we can then downstream change protein expression, gene expression, etc. So we wanted to see if that was altered as we increase our disease state, and there was no alteration. But interestingly, what we do see is that some of our complexes of oxidative phosphorylation, complexes 1 through 5, are negatively correlated with beta hydroxybutyrate lysine. So as this decreases, mitochondrial complexes increase, negative correlation. So there's some major conclusions I want you guys to be able to take away from that research. One of them is that progressing MASLD corresponds with lower serum beta hydroxybutyrate and liver HMGCS2. So what that's telling us is that with MASLD, as we get lower fatty acid oxidation, we get lower serum beta hydroxybutyrate as well too, as a result of that MASLD. This is also paired with reductions in mitochondrial turnover, such as biogenesis, autophagy, and mitophagy. So great. We know then a disease state of MASLD, we get a decrease in ketones, and that's bad for the liver overall. But if you remember before, I mentioned that ketones are utilized by other tissues. So what happens to those other tissues if we don't have them in circulation, right? Well, Dr. Taylor Kelty in our lab recently published this review article about the emerging links between non-alcoholic fatty liver disease and neurodegeneration. And there's a number of alterations that occur, but one of the things he found was that hepatic mitochondrial dysfunction can influence this disease state. And where does ketogenesis occur? In the mitochondria. So then it led to this piece of work that I was able to be a part of. That's Dr. Taylor Kelty, which is cognitive impairment caused by compromised hepatic ketogenesis is prevented during endurance exercise. So I won't dive too much into that data because frankly, we would be here all day. There's a lot of figures, there's a lot of proteomics as well too. So if you want that article, I can send it to you. But the overall take-home message that I want you guys to get is here. So what happened is we were able to knock down HMGCS2 in the liver, that rate-limiting enzyme, which then knocked down ketone bodies in our circulation. We then decreased mitochondrial function in the brain, BDNF, which is positive for the brain, decreased synaptic plasticity, and decreased cognition. So once again, showing that ketones in a disease state of MASLD are bad, but it's also bad for our brain too. So ketones are necessary for the brain. And then I think every exercise physiologist over here is like, yes, okay, exercise. We still have it. Even without those ketone bodies, we were able to maintain mitochondrial function, increase our BDNF, increase our synaptic plasticity and cognition. So once again, ketones are necessary, very necessary for the brain, but exercise is able to rescue some of that. So yay, go exercise, right? This is usually where people start to zone out, so I thought I'd give a quick liver joke. We can do a quick 7th inning stretch, get resituated in our chairs. There's probably only like 2 people in the audience that'll laugh at this, but it's fine. It's just liver jokes. Okay, so this then leads to the last study that I'm going to show you guys, and this relates to aerobic exercise, ketogenesis, and ketolysis. So the first thing I want to show you to remind everyone is that during one exercise bout of enough duration and intensity, we get a stark increase in ACAC and beta hydroxybutyrate, which I think you'd appreciate here, and then the combination of those two. This was shown back in 1983, so this is one of the first pieces of work to show this, and it's very commonly known in our exercise physiology space. I also apologize for the blurriness, but this was from 1983, so this is the best that we're gonna get, folks. So this then led to one of the studies that I'm wrapping up now. I wanted to evaluate whether chronic aerobic training increases ketogenesis in the liver and ketolysis in peripheral tissues. So we were able to take male and female rats at 6 months of age and put them on an 8-week aerobic exercise protocol on the treadmill. So for 5 days per week, we increased grade and speed for 1 hour per day. We then measured protein expression in the liver and the muscle and the brain. I Some of you may be saying, for people that are familiar with the space, like, I thought we kind of knew this already, maybe like an element of the muscle. I really want to highlight the novelty of this study. Almost all studies in ketones previously have been done in males. So this will be one of the first that actually evaluated male and females and looking at sex-specific differences in the tissue. And we're also looking at a number of tissues that are known to communicate with each other during exercise. So first we looked at our lovely liver. We looked at HMGCS2, our rate-limiting enzyme, BDH1, that enzyme that converts between ketones, and MCT1, our transporter. So HMGCS2, when we're evaluating our sedentary versus our exercise groups, our males decreased, where our females had no alteration, and females were much lower with our protein expression. BDH1, males also decreased this, females had no change, and they were naturally lower. MCT1 increased in the males slightly, and females did not change, but there was a difference between this exercise group. So exercise decreases ketogenesis in the males, but not in the females. We then evaluated a number of two enzymes from the ketolysis pathway. And hepatocytes, which make up, 70–80% of the liver, cannot use ketones. But there's a number of other cell types in the liver that I don't want to say theoretically because they can. It has been shown that they can use ketones, such as liver sinusoidal cells, Kupffer cells, stellate cells, and the list sort of goes on. So we evaluated a few elements of ketolysis, one being ACAT1, which is the conversion up here. There's no change with exercise, but female expression was naturally lower. OXCT1 was significantly increased in the females post-exercise, well, 0.53, significantly. And females had a naturally higher expression than males. So exercise increases OXCT1, a ketolysis enzyme in females. Very interesting, right? Well, I think it is. Following, we then evaluated a few ketolysis enzymes in the muscle, in the gastroc. So we saw that BDH1, this conversion between BHB and ACAC, significantly increased in the females. This is fourfold, right? You run this protein, you're like, this isn't right. I'm going to rerun this Western because that doesn't make a lot of sense to me. So you rerun it and it happens again. You're like, oh, cool, maybe a cool science finding. So stark increase in that female group post-exercise. OXCT1 and ACAT1 as well too were increased in both groups with our exercise group. So exercise increases ketolysis in the gastroc with males and females, but females have elevated conversion with BHB and ACAC. We then looked at a few markers in the brain for ketolysis. No major changes with OXCT1 and ACAT1, but we did see an increase with these males of BDH1. Females were naturally lower. So again, exercise increases the conversion between BHB and ACAC in males, but not in females. So major conclusions from that study, there's a lot going on. There's a lot of changes, so there's not just one lovely headline that I wish I could give you, at least not at the moment. Chronic aerobic exercise decreases liver ketogenesis in the males, where it increases ketolysis in the females. It increases utilization of the ketones in gastroc, but it does alter the conversion of ketones in all the tissues. Males decrease BDH1 in the liver, they increase BDH1 in the brain, where females increase BDH1 in the muscle. So major conclusions from all the data that I showed you. I know there was a lot going on, a lot of different tissues, exercise, disease state, brain. So here are the overall things that I would like you to take home with you. Progression of MASLD is associated with reductions of ketogenesis, specifically BHB and our rate-limiting enzyme. Decreased rates of fatty acid oxidation occur in rates proportional to decreased rates of HMGCS2 and beta-hydroxybutyrate. A lack of ketones can impair cognition, BDNF, synaptic plasticity, and mitochondrial function in the brain. And ketone metabolism is a large driver of chronic aerobic exercise and sex-specific adaptations. So although people used to think this may not necessarily be true anymore, right? And some acknowledgments for all this work. Thank you to Dr. Rector and Dr. Parks for obviously, even letting me participate and think of any of these studies and really led the pathway for me. Grace Mears, because without her, I think I would not know how to do anything in lab ever. She's always fixing my problems. I really appreciate her. Dr. Taylor Kelty and Dr. Renee Dashik for letting me ask as many questions in lab as possible, no matter how naive it may be. Dr. Mary Moore for always being a sounding board. Christopher Taylor for putting up with my daily lab shenanigans and always helping me when I need help. Alexa Krause, another fellow PhD student who, again, helps my anxiety every day. And then some of the lovely undergrads, Casey, Ava, Mackenzie, Abigail, and Olivia, because without them, not even like half of this work could be done. I really, really appreciate them. And with that, I say live or laugh love. I will take any questions. Thank you. Mm-hmm. Gastroc was total, so it was not, we didn't separate fiber type, and brain was prefrontal cortex. So, I'm gonna be honest, I'm not a brain expert, but I'm just gonna say yes. Is that correct, Dr. Kelty? Yep. Okay, cool. Yeah.
So I think on the.
Oh, yeah.
Sorry. Do you wanna go back to 1 to 5?
Yeah.
So do you think that some of these findings are attributed at all to changes in body mass or liver mass?
Mm-hmm.
Where we see that there's no changes in the males, no changes in the females, including the OxyC. Is that increase due to perhaps a reduction in overall liver mass in
Mm-hmm.
females, or is it ultimately not different with sedentary exercise?
So we don't typically see that females actually have any alterations in liver mass post-exercise. Um, and what was your second question? You just want me to speculate on it?
Yeah.
Okay. Um, so yeah, this could be just because females are so much smaller than males. That's always a possibility. But that's also, I'm like, well, we normalize the protein expression. Like it's all the same protein when we put it through the Western blot. You know what I mean? So it's hard for me to say yes or no to that, but that is a very good point. I will say this finding to me is very interesting. So in Masled, females are less susceptible and they're less likely to develop fibrosis. Two cell types that really contribute to that are stellate cells and Kupffer cells, both of which have been known to use ketones, and there seems to be some sort of positive benefit as more and more literature comes out. So it makes me sort of guess or theorize that maybe part of the reason why females are so protected from developing that could be part of this enzyme. So, does that answer your question? Okay. Yeah. Any other question?
I wanted to expand this slide. So this is incredible. And to keep up with it.
Mm-hmm.
How, so were concentrations in the males and females different after in the blood?
Yes. So we looked at a few different time points. We looked at an hour post exercise and we looked at 24 48 hours afterwards. I know 1 hour post exercise was different, but I don't remember 24 hours post. Yes, but I don't remember which direction. Mm. Yeah. I think it also sort of comes back to the intensity aspect as well too. So this is also a point of, so females typically have more type 1 fibers, and research has indicated that expression of BDH1 is higher in type 1 fibers than type 2 fibers, so it could be an aspect of that as well too, and less about like metabolism aspect of the whole system and more just fiber type specificities. Yeah, so. Mm. Oh, thank you. Yeah. It was treadmill. So it was treadmill running. Yeah. Mhm. Yes. Yeah. So they did not. Yeah. That was all controlled for, if that makes sense. So, yeah.
70% of the
Mhm.
males struggle a lot with the females.
Just naturally, yeah.
Much smaller, they run much better.
Mhm.
I think this is because they were exercising at a higher level.
Yeah.
I think that, but
Mhm.
their ketones are higher after acute exercise
Mhm.
than the males. Right, they were 1 hour just like.
Yeah. Yeah.
And were they on chow?
Standard chow, yeah. This is like a healthy phenotype, yeah. Any other questions? Sure, go ahead. Mm-hmm.
Is there any benefit to that population?
Yeah, the question is there any benefit with like the ketone ester, the ketone salt, in terms of masled? So yeah, Mary Moore actually did a study in rodents where she gave a dietary ketone ester, and it was very beneficial actually in terms of NAFLD outcomes. In terms of humans, my understanding is there has not been like a controlled ketone ester study yet. I'm sure it will be soon. So, yeah. Yeah, so like the shape of them, the form, whether like R versus S when we start getting into like biochemistry, um, and outcomes can be different as well too. Um, I don't know the cellular mechanisms, but it appears that there are differences in terms of response to them. So like a ketone ester is obviously different than just a ketone by itself. So does that answer your question? Potentially. We don't know yet. I always just say just exercise, but so, well, great.
Something else, you get 20 minutes back and let's say great.